What the fuck is up, you degenerates? Welcome back to the motherfucking deep dive, where we take a shitload of information and cram it down your throats.
Sounds delightful.
Today we're going to be talking about some seriously mind-blowing shit. CRISPR.
Here we go.
Yeah, you heard right, that gene editing stuff. That sounds like it's straight out of a goddamn sci-fi movie, but hold on to your asses, folks, because this shit is real.
It is.
And it's about to get fucking weird. We've got a badass expert on the line who's going to break it all down for us. So without further ado, let's cut the crap and get this shit show started.
Let's do it.
All right, motherfucker, let's dive right in. First things first, what the fuck is CRISPR anyway? You know, for those of us who don't spend our days jacking off to DNA strands.
Well, CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats,
Jesus Christ.
is a revolutionary gene editing technology. It allows scientists to make precise changes to DNA sequences. It's kind of like using a molecular scalpel.
So we're talking about literally rewriting the goddamn code of life. That's some heavy shit right there.
Yeah, you could say that. It's based on a natural defense system found in bacteria. These little bastards have been using CRISPR to fight off viruses for millions of years.
Bacteria, those microscopic motherfuckers are packing heat. That's pretty badass. Tell us more.
Essentially, bacteria use an enzyme called Cas9, which acts like a pair of molecular scissors to cut DNA at specific locations. This allows them to disable invading viruses. Scientists have figured out how to harness the system and use it to edit genes in a wide range of organisms, including humans.
So we're stealing bacteria as weapons and turning them against, well, everything. That's some ingenious shit right there.
Pretty much. And it's a hell of a lot more precise than the older gene editing methods. Before CRISPR, scientists were using clunky techniques like ZFNs and TALENs, which were like trying to perform surgery with a goddamn chainsaw.
Those acronyms give me a fucking headache. I'm glad they came up with something better. So CRISPR is like going from a rusty old tractor to a goddamn spaceship.
You could say that. CRISPR is more precise, efficient, and way cheaper than those older methods.
All right, so CRISPR is the new hot shit, but what the fuck can we actually do with it? What are the practical applications of this technology?
Well, the applications are pretty much limitless. Let's start with disease modeling. Scientists can now use CRISPR to create animal models of diseases like cancer, Alzheimer's, or even fucking HIV, which can help us understand those diseases better and develop new treatments.
Hold on, so you're telling me we can create tiny lab rats with Alzheimer's so we can test out new drugs on them? That's kind of fucked up, but also kind of cool.
It's not about being cool or fucked up. It's about scientific progress. It allows us to test new therapies without putting humans at risk.
All right, fair enough. But let's talk about those therapies. Can CRISPR cure diseases? Like, are we on the verge of eradicating cancer and all that other nasty shit?
There's definitely potential. There are already clinical trials going on for things like cancer, HIV, and genetic disorders like sickle cell disease.
So they're actually injecting people with this CRISPR stuff. That's both terrifying and exciting as fuck.
It's cutting-edge science. For example, in cancer research, they're using CRISPR to modify a patient's immune cells to make them better at attacking cancer cells.
So it's like giving your immune system a goddamn superpower upgrade. Sign me the fuck up.
In a way, yes. And with HIV, CRISPR could potentially be used to remove the virus from infected cells, something that's never been possible before.
Wait, so we could actually cure HIV? That would be a game changer for millions of people. But isn't HIV like a sneaky little bastard? Doesn't it hide in your DNA and shit?
Exactly. HIV is a retrovirus, meaning it integrates its genetic material into the host's DNA. CRISPR could potentially target and remove those viral genes. It's a huge challenge because you have to eliminate the virus in virtually every cell, but the possibility is there.
All right, that's some mind-blowing shit. But enough about diseases. Let's talk about the really freaky stuff. Designer babies. Is that actually a possibility with CRISPR? Like, can I order up a kid with blue eyes, a six-pack, and a genius-level IQ?
That's where things get ethically complicated.
Oh, come on, Doc. Don't give me that politically correct bullshit. I want the raw truth. Can we create designer babies or not?
While the technology theoretically exists to modify genes in embryos, it's incredibly controversial. We're talking about making changes that could be passed down to future generations with potentially unknown and irreversible consequences.
Okay, I get it. Messing with the human germline is a big no-no. But let's move on to something a little less controversial. What about other applications? Can we use CRISPR to create, like, giant tomatoes or glow-in-the-dark puppies?
CRISPR's applications extend far beyond medicine. It's being used in agriculture to create crops that are more resistant to pests and diseases, to increase yields, and even to enhance nutritional content.
So CRISPR could help us grow more food and feed a hungry planet. That's actually pretty fucking impressive.
Indeed. For example, researchers are using CRISPR to modify plant genes to increase their tolerance to drought, reduce their need for fertilizers, and even boost their nutritional value.
Wait, so we could potentially have crops that can thrive in shitty conditions like those dry-ass deserts in Africa. And food that's actually good for you? That's some next-level shit right there.
The possibilities are vast. And as for your glow-in-the-dark puppies, while that specific application might be a bit far-fetched, CRISPR has been used to create animals with modified traits, like pigs that are resistant to certain viruses or sheep that produce more wool.
Damn, this CRISPR shit is starting to sound like something out of a goddamn comic book. But okay, let's get back to reality for a second. I know there are some ethical concerns with CRISPR, right? Like, isn't everyone freaking out about designer babies and the potential for misuse?
You're goddamn right they are. The ethical concerns surrounding CRISPR are huge. People are worried about everything from unintended consequences to the creation of a genetic underclass.
Yeah, I can see why people are freaking out. It's like, what if some rich asshole decides to create a race of super intelligent babies and the rest of us are stuck being their goddamn slaves?
That's a bit of a dystopian scenario, but it highlights the potential for inequality and discrimination. The ability to manipulate genes raises a host of ethical questions about who gets to decide which genes are edited and for what purpose.
That's some scary shit right there. We need to make sure this technology doesn't fall into the wrong hands.
Absolutely. There are ongoing discussions and debates about how to regulate CRISPR and ensure its use responsibly.
All right, let's end this part of our deep dive into CRISPR right there. We've covered a shitload of ground from the basics of gene editing to the potential applications and ethical concerns. In the next part, we'll delve even deeper into this crazy shit.
Get ready for round 2, you degenerates.
Stay tuned, motherfuckers. We'll be right back.
Welcome back, you bunch of degenerates. Ready for some more CRISPR madness?
Hell yeah, Doc. Let's get back to this crazy shit. We were talking about all the different CRISPR systems. Like, what the hell does that even mean? Is Cas9 the only game in town?
Cas9 might be the rock star, the one everyone's heard of. But trust me, there's a whole goddamn family of these Cas proteins. Each one has its own quirks and talents, kind of like a band of molecular assassins.
So CRISPR's like a rock band, and each Cas protein is a band member. I like the analogy. Give us the lineup, Doc. Who are these other players and what the hell do they do?
Well, there's Cas12, this lean, mean editing machine. It's smaller than Cas9, so it's easier to work with in some situations.
Okay.
Plus, it has this cool trick where it can cut DNA and leave behind a sticky end.
A sticky end?
Oh, yeah.
Sounds kind of kinky. What the fuck is that?
Think of it like puzzle pieces. Those sticky ends make it way easier to insert new DNA sequences into the genome. It's like having a custom-fit piece that snaps right into place.
So Cas12 is like the surgeon of the CRISPR band, making precise cuts for easy gene insertions. What about the other band members?
Then you've got Cas13, the rebel of the group. This one doesn't even touch DNA. Instead, it targets RNA, which is like the messenger molecule carrying instructions from DNA to the protein-making factories in the cell.
Hold up. You're saying Cas13 is like the DJ, remixing the RNA instead of messing with the original DNA tracks. That's pretty damn clever.
Exactly. Cas13 is a powerful tool for controlling gene expression. You can use it to silence certain genes or even edit RNA directly. It's like fine-tuning the volume on specific genetic instructions.
Damn, CRISPR's starting to sound like a goddamn orchestra with all these different players.
Mm.
What else you got, Doc?
Well, there's this new kid on the block, Casmina. This little dude is only half the size of Cas9, making it even easier to deliver into cells. It's like the prodigy of the CRISPR world.
Casmini, the pint-sized powerhouse. That was pretty badass. But with all these different Cas proteins and techniques, there must be some challenges, right? Like, isn't this shit still experimental?
Of course there are challenges. This isn't some sci-fi fantasy where we can just wave a magic wand and rewrite the goddamn book of life. One of the biggest hurdles is what we call off-target editing.
Off-target editing? That doesn't sound good.
It's when the CRISPR system accidentally cuts DNA in the wrong spot. It's like trying to hit a bullseye and ending up shooting your neighbor's cat instead.
Jesus Christ, that's a fucked up analogy, Doc.
Mm-hmm.
But I get it. So how the hell do we prevent these off-target screw-ups?
Scientists are working their asses off to improve CRISPR's accuracy.
Okay.
They're designing new Cas proteins with better targeting and developing algorithms to predict and minimize off-target edits.
So it's like upgrading the CRISPR software to get rid of the bugs.
Exactly. We're constantly refining the technology to make it safer and more precise.
Yeah.
But another big challenge is delivery. How do we get this CRISPR shit into the cells where it needs to do its job?
That's where those viruses come in, right?
Yep.
Like using them as Trojan horses to sneak the CRISPR stuff into the cells.
You got it. Viruses are masters at getting inside cells. Scientists can modify them to carry the CRISPR components and deliver them right where they need to go.
So we're fighting fire with fire, huh? Using viruses to cure diseases. But what if those viruses go rogue? That seems kind of risky.
There's always a risk, but scientists are developing modified viruses that are safe for therapeutic use.
Right.
They strip out the harmful parts and replace them with the CRISPR components. It's like turning a virus into a FedEx truck for gene editing.
That's one hell of a delivery system.
Yeah.
But okay, let's switch gears for a minute. We've already talked about designer babies, but I bet there are other ethical concerns with this CRISPR shit. Right?
Yeah.
Like,
what about gene doping in athletes?
gene doping is a legitimate concern.
Yeah.
Imagine athletes using CRISPR to enhance their muscle growth, endurance, or even recovery. It would be damn near impossible to detect, and the long-term consequences are unknown.
Holy shit, we could have athletes breaking world records left and right and no one would even know they're cheating. That's a fucked up scenario.
Yeah.
But let's be real, Doc. Wouldn't you be tempted to use CRISPR to juice yourself up if you were a professional athlete?
Tempting.
Yeah.
Maybe. Stupid. Absolutely. We're talking about messing with the fundamental blueprint of the human body. The potential for unintended consequences is huge. All right, point taken. But let's move on from the dark side of CRISPR for a second.
Sure.
What about the positive applications? I read somewhere that CRISPR could be used to save endangered species. How the hell does that work?
CRISPR could be used to edit the genes of endangered species,
Right.
making them more resistant to diseases, helping them adapt to changing environments, or even potentially bringing back extinct species.
Hold up. You're telling me we could bring back goddamn dinosaurs with CRISPR? That's some Jurassic Park shit right there.
Bringing back extinct species is a lot more complicated than just editing a few genes, but CRISPR could certainly play a role in preserving biodiversity.
Okay, so maybe no dinosaurs just yet. But saving endangered species is still pretty damn cool. What about other applications? Can we use CRISPR to fight climate change?
Scientists are exploring the use of CRISPR to develop climate-resilient crops, enhance food production, and even engineer microbes that can capture carbon dioxide from the atmosphere.
So CRISPR could be our secret weapon against climate change.
Mhm.
That's pretty fucking badass.
It's a powerful tool with enormous potential, but it's not a magic bullet.
Right.
We need to use it responsibly and in conjunction with other approaches.
Right. CRISPR isn't going to solve all our problems overnight, but it's definitely a game changer. All right, that's it for part 2 of our CRISPR deep dive. We've explored the different types of CRISPR systems, the challenges, and some of the crazy applications.
Get ready for the grand finale, folks. We're about to get into the really mind-blowing shit.
Stay tuned, motherfuckers. We'll be right back. All right, you sick fucks, welcome back to the final round of our CRISPR deep dive. We've talked about designer babies and gene doping, but what other crazy shit keeps the ethics folks up at night?
Well, let's revisit those designer babies for a second. The idea of using CRISPR to enhance human traits, things like intelligence or physical appearance, is a goddamn minefield.
Yeah, who the fuck decides what's an enhancement? What if only rich assholes can afford these genetic upgrades? We'll end up with a world straight out of a dystopian sci-fi flick.
Exactly. We could end up creating a new form of eugenics, where the wealthy elite engineer their offspring to be superior.
Right.
That's a recipe for social disaster.
Damn, that's some serious shit. Okay, so designer babies are off the table for now. What about gene doping? Is that really a thing, or is it just science fiction bullshit?
Gene doping is a real concern, especially for athletes looking for an edge. Imagine using CRISPR to enhance muscle growth, increase oxygen uptake, or speed up recovery. It would be the ultimate undetectable performance enhancer.
Holy shit, that would change the entire fucking game.
Yeah.
We'd have athletes shattering world records left and right, but wouldn't that be, like, totally unfair? And what about the risks?
The long-term effects of gene doping are unknown. We're talking about tinkering with the human genome, and there's no telling what could go wrong. It's like playing Russian roulette with your DNA.
Yeah, that's not a gamble I'd be willing to take. Okay, so gene doping is out, but let's switch gears for a minute. I read somewhere that CRISPR could be used to save endangered species. That sounds pretty fucking amazing.
It does have incredible potential for conservation. Imagine using CRISPR to make endangered species more resistant to diseases or to help them adapt to changing environments.
Wait a minute, are you telling me we could bring back the goddamn woolly mammoth with CRISPR? That would be some next level shit.
Bringing back extinct species is a complex issue,
Yeah.
but CRISPR could definitely play a role in preserving biodiversity and protecting endangered species.
All right, so maybe no mammoths just yet. But saving endangered species is still pretty badass. What about other applications? Can CRISPR help us fight climate change?
Scientists are exploring the use of CRISPR to develop climate-resilient crops, increase food production, and even engineer microbes that can capture carbon dioxide from the atmosphere. It has the potential to be a game changer in our fight against climate change.
Damn, CRISPR really does sound like a superpower.
Mhm.
But with all this power comes a shitload of responsibility, right?
Absolutely. CRISPR is a tool, and like any tool, it can be used for good or evil. It's up to us to decide how we want to wield this power.
Well said, Doc. I think our listeners are starting to get a sense of just how powerful and potentially transformative this technology is.
We've only scratched the surface in this deep dive. CRISPR is a rapidly evolving field with almost limitless possibilities. It's both exhilarating and terrifying.
You're telling me. This shit has blown my fucking mind. But okay. I think it's time to wrap things up.
Finally.
Hey, fuck you, Doc. All right, folks. That's it for our deep dive into the wild world of CRISPR. We've covered a lot of ground, from the basics of gene editing to the potential benefits and the ethical nightmares. We've talked about curing diseases, enhancing crops, designing babies, and even bringing back extinct species. CRISPR is a technology that has the potential to reshape our world in profound ways. It's some seriously mind-blowing shit, and we've only just begun to explore its possibilities. So stay tuned, motherfuckers, because the future is going to be one hell of a ride.
Buckle up.
Yeah, hold on to your asses. Catch you later, motherfuckers.